Just like we did microarray normalization, RNA-seq also needs normalization. Why there is variation within the data? Sometimes we see that different sequencers, they produce different read outputs. Sometimes there may be variations among different lanes of the sequencers. And while we are assigning these reads or putting those reads, we're using some alignment softwares into different genes, we observed that the genes which are having, or the transcripts which are having the longer lengths, they will get more reads. So we need to take into account all these factors and normalize our data so that we can do our within the experiment comparisons, and we can compare the same the expression of the same gene across different treatments. So RNA-seq has some challenges. There is uniformity of sequence coverage. So the sequencing reads which are coming from different regions of a genome or a transcriptome, are they uniformly derived from each area? So that is an important aspect to cover. The quantity of sequence required to reliably detect RNAs of lower abundant classes, that might also be a big issue. So what quantity of the RNA we should require so that we can capture the genes which are low expressed? So where you define the threshold, that is the important challenge. The quantification and conversion of relative quantification to absolute RNA concentration, that is also an important aspect or a challenge. Transcriptomes of organisms with larger genomes containing genes with more complicated structures, and they also pose some special challenges to this analysis. The read counts will be higher if sequencers they produce more reads, and we have said earlier that longer genes will get more reads hit to them, so we need to account these mapping biases. So here we see two genes. Gene A has two alternative splice variants, so it's represented as gene A and A.1. Then there is a gene B. If you look into the lengths of these two genes, we see that B is quite longer as compared to the two alternative splice variants of A. So there is a lengthwise biasness. So obviously the longer gene will get more reads aligned to it. So we need to take into account this factor. So if you look into here, for example, A has a length of 1 kb, or B has a length of 2 kb, while comparing A and B at this using these raw counts is not a fair deal. So in second way, if you look into these treatment 1, treatment 2, treatment 3, these are all different samples. So they have been sequenced using maybe different lanes within the sequencer. So we can have some summation of total reads coming from here. For example, 38 here. These may be 44 plus 37 and so on. So we have overall different numbers of reads which are there as far as these samples are concerned. So different samples coming from different lanes. So if we are comparing the same gene across different treatments, for example, we are comparing gene 1 in treatment versus these wild types. So we need to take into account this factor that these numbers 23, 44 and 7 and these 220, 15 and 33, they are coming from different pool of reads. So sequencers have lanes, and lanes they might have variability as far as producing the overall number of reads. So if more reads are produced within a lane, more reads will be assigned to those genes. So we need to take into account both these factors while normalizing our data. So Murtazvi et al., they provided this normalization method. We call it as RPKM. It stands for reads per kilobase of transcript or exon model per million mapped reads. So method for quantification of transcript levels. RPKM measure of read density reflects the molar concentration of transcript in the starting sample by normalizing for RNA length and for the total read number in the measurement. It facilitates transparent comparison of transcript level both within and between the samples. Within sample means, for example, in gene A and B within treatment 1, and when we talk about across the samples, so we have, for example, the comparison of same gene across different treatments. So RPKM is represented by this formula. So it is number of reads mapped per gene length in kb, per total reads in that samples in millions. So we take the read numbers, we convert them into the millions. So in this way, every number is coming from 1 million. So it's kind of we are fixing for the variation within the total within the samples as far as the total number of reads assigned to each sample by sequencer. So here we have C, which is the count of the mapped reads or raw counts. L represents length of the transcript. M represents map reads. So RPKM is given by C, the raw count, for example, number like 23 or something, which we have seen in the earlier count matrix, divided by length of that transcript divided by 1000. So that is the length in actually 1 per kb, we can say the length in kilo base pairs, kbs. We take the total reads, whatever those number of reads were present in that sample, and we divide it by this million, this number. So that's why we will get the read number in millions. That M stands for the mapped reads. We are not looking into the whole reads which are present in that sample, but the mapped one. We can change this equation into this pretty easy to calculate equation. We can take the number 1000 and million and multiply them and bring them up in that equation. So we can use this relation which is there. So it is C into 10 power 9 divided by L into M. Let's understand this by solving this example. What is the RPKM for a transcript of length 2500 kb with 900 alignments in a sample of these 10 million reads, out of which 8 million reads were mapped. So 8 million reads were mapped. Which figure is important? 10 million or 8 million? Obviously we are looking for mapped reads, so 8 million is important. This number we use in our calculations. 900 alignments here is the 900 reads which are hitting to these particular gene, whose length is 2500 kilobase pairs, kbs. So we can solve it. So it's 900 into 10 power 9 divided by 2500 into 8 million. So 8 million can be written as 8 into 10 power 6. So it is 4.5 though. So the RPKM number is the normalized count number is 4.5. The raw number was 900. So this 4.5 number we can compare within the treatment and across the treatments. So here we do another calculation. So we are asking here how many reads are required to map at 1 RPKM with a transcript of 2 kb length from a total of 40 million mapped reads. So here we are interested in how many reads are required to map. So we are actually looking for C here, plug in different numbers. We do some calculations and we came up that we need 80 reads to be hit with this transcript of length 2 kb, whereas total 40 million reads they were mapped for the whole sample. So we need 80 reads to be mapped with this in order to get a RPKM of 1. Shrapnel et al., who wrote a software called as Tophat, which is very famous in this RNA-seq analysis, where they provided another normalization technique, a slight variation, though the concept is same, but they are using different words. So they say fragments per kilobase of transcript per million mapped reads. Rather than RPKM, it's now FPKM. So this is applicable where we have paired and RNA-seq experiments. They produce 2 reads per fragment. So they take an RNA fragment and make 2 reads. Sometimes FPKM counts fragments here in this example. So they are not counting actually reads. So and in some situations both reads they might not map. If we count the reads, that might double the counts of those sum of the fragments. So in order to compare these two situations, or in order to control that lingo, they use the word fragments per kilobase of transcript per million mapped reads rather than RPKM or reads per kilobase of transcript per million mapped reads. Sometimes, most of the times while we are using those differential gene expression studies, we are taking the same gene and we are comparing across different treatments. So here some scientists are of the view that we should take this K out from this equation We should not normalize for the gene length since we are comparing the same gene across different treatments, so it is unnecessarily reducing the number, or the count number will go small if we are using RPKM. So rather than, we should use RPM. So simply take the reads, mapped reads, and divide that by total million of the mapped reads. So that's why it becomes RPM. So that is count divided by M over 1 million, or we can simply rewrite it as C into 10 power 6 by M. So in RNA-seq, the normalization is necessary because we need to take into account different variations within the gene lengths as well as the number of reads produced in the sequencers.